Thank you for your presentation. Thank you to everybody. Very young people. I'm very happy to do this. Okay. Thank you to Lobo to invite me. And this is the first time that I visit Slovakia and Bratislava, and I was very excited to come here because when they told me that there are so young people, so young students, I was very happy to come because I think that the future is yours. So, and also because I have 3 children, so more or less they have your age. So one of them, sure. Okay. Today I will speak about 2 projects. The first project regard Leonardo DNA project. I tell you this is a project that is still in progress because we are looking for the DNA of Leonardo da Vinci. I came from Florence, and Florence is very close to Vinci. This is a little village near Florence in which Leonardo born more or less 500 years ago. And the second project is a very interesting project because we analyzed the DNA of Pompeii victims. You know where is Pompeii? Pompeii is a very important site in Italy. It was destroyed by the Vesuvio, okay, by the eruption in Vesuvio in the 1st century after that. And we present the first result that we published this last year on this journal, Current Biology, and about the cast. We analyze the DNA extract from the cast. So this is very interesting. Okay, we start with Leonardo da Vinci project. This is a project that, in collaboration with other colleagues from other parts of the world, especially from US, there is Jesse Osbel. He's professor at the Rockefeller University. Is the coordinator of this project. The coordinator, but he doesn't coordinate the DNA because my group coordinates the DNA. But there are also other colleagues that work on painting of Leonardo and on other issues of Leonardo. Leonardo da Vinci DNA project aims to, as right here, to establish whether the remains believed to those Leonardo da Vinci Adambart's castle in French belong to Leonardo or not. And to do this, we need a marker, a DNA marker that we can use for establish if these remains belong to Leonardo. And how is possible to have a DNA marker that permit us to establish if these remains belong to Leonardo? Well, it's very interesting because it's very interesting. On your right, there is handmade the Leonardo da Vinci genealogic tree. This is very amazing because the yellow, the person highlighted in yellow, this one, okay, this one, are still alive. And we try to recover the DNA from this. But as you can imagine, Leonardo is here, and Leonardo has a lot of brothers, more or less 24 brothers. So yes, his father is very important man, and with a lot of women. So as you know, there is a particular fragment of the DNA, the Y chromosomes, that it has inherited by man to child, to other child. The important is the child is a male. So for this, we are looking for the Y chromosome of Leonardo. And we start with this, okay? Here you can see Caterina is the mother of Leonardo, Ser Piero is the father of Leonardo, and this is Leonardo. there are 20 generations to arrive at live decisions. But what we have to do is first, oh my God, what happened? Okay. Is to analyze the DNA, the Y chromosome of live descendant. But this is not enough, because we go in the time to analyze the Y chromosome of the generation before the live descent and the generation in which Leonardo lives. So this is very important to do this, because you know that usually the genealogy is not the same of biology. Sometimes happens that the mother, of course, we know who is the mother, but the father not always is the same that we know. So yes, this is true. And the only thing that we can do is go to sampling people who live in the same times of Leonardo and follow the genealogy. So this means to go to collect several samples along this genealogy. So ancient bones and oral swab from living donors. So this means that we start with, of course, the more easy things. So the more easy things is the oral swab for the modern decade. And we try to collect some ancient sample, especially Petrus bone. You know what is the Petrus bone? The Petrus bone is a bone here, okay? Is here. And it contains a lot of DNA. So it's more easy to collect DNA from these bones because it contains a lot of DNA. Its DNA is not so degraded, so it's very easy to analyze. It's more easy than teeth or other part of the bones, of the skeleton, of course. And this is the Petrus bones, okay? The Petrus bones, the part of the Petrus bones important for our study is the part C. So we have to cut these bones, we have to collect the bone powder from this, and we start the analysis. But, as you know, we start with the modern descent. So the modern descent are these in this slide: 1, 2, 3, 4, 5, and 6. And we collect the DNA from the modern descent. Modern descent are, fortunately, in these three, all male, and it was possible to do this. Here, this is very private pics that I show you, are the pics of the modern descent that now, of course, I brushed his face because it's important, but with oral swab, we collect, this is my colleague, this is Professor Elena Pilli, she's a molecular forensic anthropologist, and we collect 3 years ago, no, I forgot, the DNA from them in order to verify if they have the same Y chromosome. Because they have the same Y chromosome, because they belong to the different line, different type they college here in Tommaso, and you can see, you can arrive, you follow this line, to the Y chromosome of Leonardo. So this is the first step of the study. So we have to demonstrate that different lines have the same Y chromosomes. If you understand me? Yeah? Okay, perfect. Okay, these are the results. We use a normal forensic kit, okay? The Power Packs. This is a kit for the Y chromosome. And it's very simple to see that all the people that we have investigated have the same Y profile, so have the same Y chromosomes. So they have the same Y chromosome, even they belong to different lines. This is very important. This is very important results. Now, the second step of these studies is to verify if some people who live contemporary to Leonardo or are ascendant of Leonardo have the same Y chromosome of the descent. So we start to analyze this. So this is Tommaso. Okay, he lived in 17th century, and this descent have the same Y chromosome. So this means that mother and father work well. Okay? Okay. Well, now we decide with some colleagues to go very far in time. And we try to identify two very close relatives of Leonardo. This is uncle, and this is the grandfather. Okay, the historical document tell us that both probably were buried in Vinci, in this little village near Florence, in the church. The church is Chiesa di Santa Croce in Vinci, and this is Alessandro Avezzosi and Agnese Sabato, with the genealogic tree in her hand. They produce the research that permit us to see that in Vinci, probably under the floor, there are some tombs in which are buried the grandfather and the uncle of Leonardo. So, more or less 2 years ago, we started this excavation in Vinci Church, and it was surprised when in the floor appear these tombs. This was a tomb inside this church, and in these tombs there are present some bones, but these tombs unfortunately was very presented this excavated because there was some work on the floor. So this not the skeleton was in connection, but we was lucky because in this church, in this tomb, we found two Petrus bones. Okay, one of these is US7, and the second US7, number 2, and arbitrary ossicle. We found this because it's possible to extract the DNA also from this. Of course, before to start the DNA analysis, we decided to use the data with the radiocombat dating. So we did these bones. And the results was very interesting because the first bones was very ancient, 657 after that, and the second bones, this one, was contemporary where the uncle of the grandfather lived, because it was 4021, 4057. So very, very, very a period very close to the way when the ascendant of Leonardo lives. So what we do, of course, we analyze them with the NGS technologies. So I want to bore you, of course, with the technologies now. But the NGS technology is a new technology that permits us to recover a big amount of DNA. And especially it's important because when we work on ancient DNA, if you use this technology, you have the possibility to discriminate between endogenous and exogenous DNA. So this is very important because the contamination is a principal neighbor, a principal problem that when we start and we study the ancient DNA. So we use these technologies. We analyze the DNA, and we see there are some endogenous DNA with good preservation. We did the sex determination, and with this methodology, the twist capture, we go to capture the DNA, the Y chromosome of this of course. We demonstrate that they belong to the male, but they was two male, okay? Well, of course, we analyze three bones, and we know that the other two are not consistent with the data, but we analyze three. And we see that endogenous DNA is very good in both the previous born, okay? So after this, we go to determine some other results that permit us to see that the DNA was ancient and authentic. This is another slide that shows us this, okay? And after this, we we determine the sex, and we see that both individuals were male, especially the US, this one, US A2, the individual that have the radiocarbon dating good, was a male. And we start with the twist capture, and we analyze the Y chromosome haplogroup. And unfortunately, the US Petrus-Bone haplogroup, the Y haplogroup, was this R1B1B2A, that it was different between the haplogroup, the Y haplogroup of the descendant. So the mystery is still open because we have to do another excavation in Vinci, because probably this individual was not the ascendant of Leonardo, or the descendant of Leonardo before Tommaso. Sometimes they happen, and the Y chromosome change, but we don't know. So for this motive is still in progress. But okay, this is not a good slide, but some colleagues analyze some documents of Leonardo. One letter, the Holy Child, the other artwork of Leonardo, and they find in the analysis the same haplogroup A1B1 that we find in the descendant. But as you can see in the previous slide, this Y haplogroup is more frequent in Italy, 10% and 20%. So it's very difficult to demonstrate that the Y chromosome that we recover from the Holy Child or the letter of Leonardo, etc., are authentic. And now we are currently conducting a detailed analysis of the shared Y chromosome. So what we need in the future? In the future, we need to discover other tombs, for example, of this individual, and other tombs in Vinci. And I ask you to stay tuned, because probably will be some good news in the next future, okay? This is the Leonardo DNA project, and this is the true story of the Leonardo DNA project, okay? This is the first part of my talk. Now, I want to speak about Pompeii. Because I want to speak about Pompeii. Pompeii, as you know, is a very important archaeological site. And 3-4 years ago, I received a big amount of money, more or less 1.5 million euros to produce a study on Pompeii. The name of the project was Pompeii Molecular Portrait. Do you know what is this, a portrait? Is a photo, a pics, okay? Because it's possible with this project to reconstruct the genetic variability, the genomic variability of the inhabitants of Pompeii. Because with eruption, several people died there, and it's possible to analyze their DNA and verify how is the genetic variability in ancient Roman site, town, in the first century after that. So we analyze 200 individual, and now the paper is under review on Nature, okay, because we are publishing these studies on Nature. But we present today a very interesting, for me, I think, study, because with this result that we obtained from the cast, now I'll show you what does it mean, cast. We have the possibility to rewrite the history, okay? Because this is very interesting. So, these are cast. These are victims, okay, that are in Pompeii. The cast has very amazing things because under the cast there are bones, and the cast was built by some archaeologists that put some substance in the ground, and we create this cast. Okay, this cast is very interesting because give us a moment very not happy, of course, the moment before they die. So, but this is very impressive. There are a lot of casts. This is the mystery, the villa of mystery, the house of mystery. This is another cast. You can see there is the teeth here, so it's easy to recover DNA. This is before his restoration. And in Pompeii there is a room in which are displayed all the cast that we analyze. So, other two casts. This is very famous. This is the two girls that embraced before they arrived the death, unfortunately. And, for example, we can collect the DNA from some bones that is present in the cast. So, this is another man who, of course, have fear because the very hot ash arrive here in Pompeii with the Vesuvio eruption. And look, this, it's present also the dress, the clothes, are present also the clothes. So, 3 years ago, we go there with my colleagues to sampling some individuals in Pompeii. This is Professoressa Elena Pilli, this is Alessandra Bodi, this is Stefania Vai. They are professors now in Florence, in my lab. And first of all, we decide to analyze 3 different individuals. So this one, this family, and these 2 girls. Because the archaeologists tell us that probably this come from Middle East. This person come from Middle East. Probably this was a family of recovering the house of the golden bracelet, because one of this one is supposed to be a female. She has an armilla. You know what it is, the armilla? Armilla is a bracelet. Is a bracelet with half kilos. So very important. She has this armilla at the left arms, yes. And as you can see, but I'll show you in another piece very more easy, there are 2 children, 1 and 2. So they suppose the father, 2 children, and the mother. And these 2 girls that probably sister or not, that embraced before the event of the Vesuvio eruption. Okay? This is the history. This is what the archaeology say. And I was very, very, very happy to do this because this is the first time that we are able to analyze the DNA from the cast. This is the first time because never other scientists have tried to do this during the studies of Pompeii. So this is very, very, very interesting. And we collect also other individuals, but unfortunately, when you work with ancient DNA, it's not always obtain results. And even we start with 14 individuals, only 6 we get, only from 6 we get results. And fortunately, the 6 are the peaks that I show you. Well, the workflow that we apply on 40 samples are very interesting because this is NGS workflow. We use the technologies of target enrichment capture. So a question for you. If I think that there is a family, okay, what does it mean, the family? A family means that we have a mother, a father, and 2 children, right? Okay. You know, when we speak about Leonardo, we analyze the Y chromosome. But in this case, we can analyze another molecular marker that it is mitochondrial DNA. Do you know mitochondrial DNA? What is this? Is a DNA that inherited between mother and children, okay? But it stops in the male generation. So this means that I have the mitochondrial DNA from my mother, but I can give my mitochondrial DNA to my children, okay? So when we decided to analyze the family, the first step was to analyze the mitochondrial DNA of different individuals that we suppose to be a family. And what results I expect? Well, I expect to have 3 mitochondrial DNA similar, okay, that belong to the mother and 2 children, okay, and other mitochondrial DNA different, right? It's okay for you? It's normal, no? The same for the 2 girls. If 2 girls are sisters, they have the same mitochondrial DNA, okay. Well, we was very surprised when we read the results. We see that the empty DNA haplogroup and haplotype was totally different. But the very interesting things, it was that all the people that we analyzed was male. So this is the first result that we obtained. So, House of the Golden Bracelet. The House of the Golden Bracelet, archaeologists have assumed that it was a family of father, mother, and 2 children. Paleogenomics indicates that the presumed woman, individual 52, this one. So the presumed woman, the woman who has the bracelet, okay, was a male. But the mitochondrial DNA of this family, but it's not a family, was totally different. So what probably happened? Probably happened that this is a slave that when they arrive the ash from the volcano Pompeii, they go away and they take these children that was there and bring with them the children. And unfortunately, they died. But this is not a family, okay? This is not a family. And other results we obtain as a report here, because we decide to analyze, of course, not only the sex and the mitochondrial haplotype, but we do a very important genetic analysis on the nuclear DNA. And we see that probably they come from, for example, the individual 53, this one, okay? The children here, it come from North Africa because it's possible to see the geography and the region of the people. So they are not local. For this motive, we suppose that are slaves that work in Pompeii and stay there. So the family is not the family. Okay, House of the Cryptocopticus, 2 individuals, 21 and 22. Of course, they are not sisters, but they are not female. They are two male. They are two male that stay there. And the genetic variability tell us that both fall with Mediterranean genetic variability. So they are local probably, but they are not two female. This is a very important discovery. And the third individual, this is confirmed that it was a male, and his ancestry, it was Anatolia, Asian or Eastern Balkan. And as isotope analysis indicate the value outside the range described of the Roman population, suggesting the individual may have come from different origins. So you can see we have rewrite the history of this particular house. But there are other very interesting results on Pompeii for the other house that we will read in some publication that we arrive very, very soon. Okay, okay, this is already said. And yes, when you analyze the whole genome, we see that Pompeii individuals have a very big genetic variability. So this means that, as you imagine, the Roman town was populated by different people that come from different parts of the Mediterranean and the Roman Empire. So from Northern Europe, from Southern Europe, from Eastern Europe, from Middle East, etc., etc., etc. So it was a very interesting result on 6 individuals. And as I wrote here, I repeat what I said before, that the genetic variability of this population is very high. And we know that with ancestry DNA, it's possible to reconstruct the phenotypic trait. So this means it's possible to see if people was this pale skin, the dark skin, blue eyes, etc., etc. So this is very amazing. And we try to see that, for example, the individual 25, 51, and 53 had brown eyes, and the individual 52 had dark skin, black hair. For this motive, we think that we come from the Northern Africa. Okay, so as you can see, we have the possibility to reconstruct not only the genetic variability and the geography of the people, but we can reconstruct the phenotypic trait. This is very important, especially for other parts. In some years ago, we analyzed, for example, the Neanderthal man, and we see that some of them have pale skin, red hair. So with the DNA, with ancestry DNA, it's possible to do this. For example, I have not reported here, but we have the possibility for an house. There is a portrait in this house. We found some bones in this house. We analyzed the bones of this woman. It was a woman, and we compared the DNA of the color of the hairs of the skin of the eyes that are present in this house, in this portrait, and are the same of the skeleton that we found in this house. So this is very, 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 very interesting. Well, in summary, in this project, we generate a whole genome data from skeletal material in plaster cast from Pompeii, and the data contradict the previous narratives about the victims' identities and relationships. And the Pompei individuals are primarily descendant from recent East Mediterranean and immigrants. So this is very interesting. And now we are waiting for the other genomes, 100, more or less 200 genomes that is arriving, and we are publishing about Pompei sites. So more or less we have at the end of my presentation. This is my group, okay? This is my group, this is my colleagues. First is Professor Martina Lari. She's in molecular paleogenomics. Elena Pilli, she's professor, she's a molecular forensic anthropologist. Professor Stefania Vai, she's, oh my God. She's paleogenomics. Alexandra Modi, she's a researcher. Stefania Moreira, she's a researcher. And on the second line, there are all my postdoc and PhD students. We are involved in a very, very important project. We are involved, for example, in analysis of ancient Paleolithic genomes. We are involved in ERC of last Neanderthal. We are involved in other projects. If you want to visit my lab, or if you want to come in my lab, I don't know, for your in your future, you are welcome. And I hope don't have a mistake. It's correct. It's correct. Thank you for your attention. Dakujem džabažu pozornost. Okay? Okay. I try it.